Hey, this is Zayam from Z Biology. So this is the second part of the cell cycle in which we are going to discuss the M phase. So starting off from prophase, the main phase. In prophase, actually the cell undergoes visible changes. You see the nuclear envelope that starts to disappear. We have the centrosomes which are doubled, the mitotic spindle forms. So you can see here the disappearing nuclear membrane. Rest remains the same. Now, what happens inside the nucleus? We have 46 chromosomes. All of them have 2 chromatids. That means duplicated chromosomes. Now, let's look at the steps during the prophase. The chromatin material condenses to form the chromosomes. We have visible chromosomes. The mitotic spindle forms. We can see the microtubules here, and the nuclear envelope starts to disappear. So these are the major events that are occurring in the prophase. Then let's just move on to the next phase, prometaphase. This phase is actually an intermediate between metaphase and the prophase, in which the nuclear envelope has disappeared completely and the cell starts to arrange the chromosomes. So you can see the microtubules connecting to the chromosomes here. The microtubules attach to the chromosomes, to the center of the chromosomes, called as the kinetochore, that holds the chromosomes in place, and the chromosome starts to arrange right in the center. That is the equator of the cell. So after this, what we have, the chromosomes are exactly the same. Duplicated chromosomes, 46 in number. So now looking at the next major phase called as the metaphase. In this phase, all the chromosomes are exactly arranged into the equator of the cell. That means the middle of the cell. The centrosomes attach to the chromosomes via microtubules onto the kinetochore of the chromosomes. So this here is the center of the cell, called as the equatorial plate or the metaphase plate. Now still we have 46 chromosomes, all of them duplicated. By duplicated I mean 2 chromatids in one chromosome. So metaphase is the phase in which chromosomes exactly arranged into the middle of the cell, the equator of the cell, the center, and joined to the poles by microtubules. Now moving on to the next phase, the anaphase. In this phase, the cell actually enlarges and the poles move into opposite direction. The centrioles splits the chromosomes in two like so, and now we have more chromosomes. That means we have 92 chromosomes, all normal ones. So we have to understand this a bit. Firstly, we had duplicated chromosomes. In metaphase we had duplicated chromosomes, but in anaphase we are now having 92 normal chromosomes. That means the chromosomes have split in two. That is because the centromere, they were moving in the opposite direction and they pulled the chromosomes apart. So this is what happened in the anaphase. Chromosomes split and move to the opposite poles, and the poles start to move in opposite direction. This is where we can clearly see the cell dividing. The next phase is the TLow phase. Now in telophase, the cell shrinks and actually is going to divide into two. We can see the nuclear envelope reappearing, and the cell has now two nucleuses. The same cell has the two nucleuses. Each nucleus having 46 chromosomes, that means one cell having 92 chromosomes, all the normal ones, but the nuclear envelope now reappears. We can see this cleavage here called as the cleavage furrow, which actually helps a lot in dividing these cells. So the chromosomes decondense again into the chromatic material, the nuclear envelope reappears, and the cell starts to divide into two. Next, we have the actual division called as the cytokinesis. Cytokinesis is the division where the organelles divide into two. All the organelles are equally divided into the two cells. So the cytokinesis is the division of cytoplasm. Whereas all this, the prophase to telophase, is called as the karyokinesis. So this was mitosis, the division of cell in which the cells divide into two exactly similar daughter cells.